Let us learn about blotting. Blotting is the process of transferring macromolecules like proteins, nucleic acids, etc. from a gel to the solid surface of an immobilized membrane to detect the molecules that have been transferred. This technique is accomplished by separation using electrophoresis, transfer to a membrane, detection with a labeled probe. Types of blotting techniques. So there are three major types, which includes southern blotting, northern blotting and western blotting. Southern blotting. This is an image of southern blotting. Southern blot is a method used for detection and quantification of a specific DNA sequence in DNA samples. The method is named after the British biologist Edwin Southern, who first published it in 1975. Southern blots can be used to analyze an organism's total DNA, also known as its genome, in order to identify a specific sequence of interest. Procedure. First step is DNA isolation. The DNA to be studied is isolated from various tissues. The most suitable source of DNA is known as blood tissue. However, it can be isolated from different tissues like hair, semen, saliva, etc. Second step is DNA digestion. Restriction endonucleases are used to cut high molecular weight DNA strands into smaller fragments. This is done by adding the desired amount of DNA, which can be changed according to the probe used and the intricacy of the DNA with the restriction enzyme buffer and purified water, then incubate the reaction at 37 degrees Celsius overnight. The third step is gel electrophoresis. The DNA fragments are then electrophoresed on an agarose gel to separate them by size. If some of the DNA fragments are larger than 15 kilo base, then before blotting, the gel may be treated with an acid such as dilute hydrochloric acid. This depurinates the DNA fragments, breaking the DNA into smaller pieces, thereby allowing more efficient transfer from the gel to the membrane. The fourth step is denaturation. If alkaline transfer methods are used, the DNA gel is placed into an alkaline solution, typically containing sodium hydroxide, to denature the double-stranded DNA. The denaturation in an alkaline environment may improve binding of the negatively charged thymine residues of DNA to a positively charged amino groups of membrane, separating it into single DNA strands for later hybridization to the probe, and destroys any residual RNA that may still be present in the DNA. The fifth method is blotting. A sheet of nitrocellulose membrane is placed on the top of gel. Pressure is applied constantly to the gel to ensure good and even contact between gel and membrane. 5 methods can be used to transfer DNA fragments to the solid membrane, and they are upward capillary transfer. This method transfers the DNA fragment upward from the gel to the membrane, where the flow of the liquid or the buffer will be upward. Downward capillary transfer. This method is done by placing the gel on the surface of the membrane, usually nylon charged membrane, and the DNA fragments will be transferred in a downward direction with the flow of the alkaline buffer. Simultaneous transfer of two membranes. This method is used to transfer DNA fragments of high concentration simultaneously from the gel to two membranes. Electrophoretic transfer. This method usually uses large electric current, which makes it difficult to transfer the DNA efficiently due to the temperature of the buffer used. So these machines can be either equipped with cooling machines or used in a cold area. The next method is immobilization. The membrane is then baked in a vacuum or regular oven at 80 degrees Celsius for 2 hours or exposed to ultraviolet radiation, nylon membrane, to permanently attach the transferred DNA to the membrane. Hybridization method. After that, a hybridization probe, a single DNA fragment with a particular sequence whose presence in the target DNA is to be ascertained, is exposed to the membrane. The probe DNA is labeled so that it can be detected, usually by incorporating radioactivity or tagging the molecule with the fluorescent or chromogenic dye. To ensure the specificity of the binding of the probe to the sample DNA, most common hybridization methods use herring sperm DNA for blocking of the membrane surface and target DNA, and detergents such as SDS to reduce non-specific binding of the probe. The next method, which is the last method, is the detection. After hybridization, excess probe is washed from the membrane, typically using SSC buffer, and the pattern of hybridization is visualized on X-ray film by autoradiography in the case of a radioactive or fluorescent probe, or by the development of color on the membrane if a chromogenic detection method is used. Applications of sudden blotting. Identification of a single gene in a pool of DNA fragments, gene mapping, analysis of genetic patterns of DNA, detection of specific DNA sequence in a genome, study of gene deletions, duplications and mutations that cause various diseases, detection of genetic diseases and cancers such as monoclonal leukemia and sickle cell mutations, detect the presence of a gene family in a genome, DNA fingerprinting and forensic tests such as paternity testing and sex determination. Next, we will see about northern blotting. Here you can see the image of northern blotting procedure. The technique that is used in molecular biology research to study gene expression by detection of RNA or isolated mRNA in a sample is called northern blotting or RNA blotting. It is a classical method for analysis of the size and steady state level of a specific RNA in a complex sample. Principle of northern blotting. The fundamental principle of northern blotting is to separate RNA based on their size using gel electrophoresis and identified on a cellular membrane by means of hybridization probe with the base sequence corresponding to all or a part of the chain of the target RNA. Method of northern blotting. First step is RNA isolation. Multiple ways of isolating RNA, but all have some common attributes such as cellular lysis and membrane disruption, inhibiting of ribonuclease activity, deproteinization and recovery of intact RNA. Some of the common methods of RNA extraction are RNA isolation by acid guanidinium thiocyanate phenol chloroform extraction, RNA isolation by column-based technology. Next step is separation of RNA using gel electrophoresis. RNA have secondary structure formed by intramolecular base pairing that prevents RNA from separation according to their size. Denaturing agent like formaldehyde or glyoxal or dimethyl sulfide oxide disrupts the secondary structure. Separation of RNA is better in glyoxal or DMSO system as sharper band for specific RNA is detected by hybridization. Size of RNA fragments are compared by migration distance with those of molecular weight markers. Gels are stained with ethylium bromide after electrophoresis to detect molecular markers and rRNAs. The third method is transfer of RNA to a membrane. Nitrocellulose and nylons are commonly used membranes. Nylon membrane are more durable. RNA are transferred via capillary or vacuum transfer. Vacuum transfer are more efficient but require special transfer apparatus. Electrophoretic transfer method is available only for nylon membrane due to low concentration of salts required to bind the RNA. Transferred RNA are then immobilized to membrane by baking at 80 degrees Celsius or through UV cross-linking in case of nylon membrane. Hybridization and washing. Hybridization is performed using radio or fluorescently labeled probe to identify specific RNA immobilization. Pre-hybridization blocks single-stranded probe from binding on non-specific sites on the membrane. Hybridization solution should contain 50% formamide to ensure hybridization at lower temperature and minimize RNA degradation. The membrane is washed in the buffer containing lower concentration of salt to remove excess probes. The next step is visualization. Detection of specific transcript through autoradiography. Membranes are placed over X-ray film. The X-ray film darkens where fragments are corresponding to the radioactive probes. Applications of Northern blotting. Standard for studying and inspecting gene manifestation design between tissues, organs, developmental stages, pathogen infection and over the course of treatment. Diagnosis of several diseases like Crohn's diseases including viral infection. Exhibit overexpression of oncogenes and downregulation of tumor suppressor genes in cancerous cells. Study of RNA degradation and splicing. Detect specific mRNA molecular weights and contents in a sample. Identify mRNA produced by the transgene to protect recombinance. Next we will see about Western blotting method. Here there is an image of Western blotting workflow. The Western blot, sometimes called the protein immunoblot or Western blotting, is a widely used analytical technique in molecular biology and immunogenetics to detect specific proteins in a sample of tissue homogenate or extract. Besides detecting the proteins, this technique is also utilized to visualize, distinguish, and quantify the different proteins in a complicated protein combination. Western blot was introduced by Taubin et al. In 1979. For the accomplishment of the Western blot, there are three elements: separation of proteins by size, transferring proteins to a solid support, and marking proteins by primary and secondary antibodies for visualization. Procedure or steps: extraction of the protein, gel electrophoresis using SDS page, blotting using electrical or capillary blotting, blocking using bovine serum albumin, treatment with primary antibody, treatment with secondary antibody, that is enzyme labeled anti-antibody, treatment with specific substrate. Enzyme is alkaline phosphatase. Substrate is p-nitrophenyl phosphate, which give color. The first step is extraction of protein. Cell lysate is most common sample for Western blotting. Protein is extracted from cell by mechanical or chemical lysis of cell. The step is also known as tissue preparation. To prevent denaturing of protein, protease inhibitor is used. The concentration of protein is determined by spectroscopy. When sufficient amount of protein sample is obtained, it is diluted in loading buffer containing glycerol, which helps to sink the sample in well. Tracking dye like bromothymol blue is also added in sample to monitor the movement of proteins. The second step is gel electrophoresis. The sample is loaded in a well of SDS page sodium dodecyl sulfate polyacrylamide gel electrophoresis. The proteins are separated on the basis of electric charge, isoelectric point, molecular weight, or combination of these all. The small size protein moves faster than large size protein. Protein are negatively charged, so they move towards positive anode pole as electric current is applied. The next step is blotting. The nitrocellulose membrane is placed on the gel. The separated protein from gel get transferred to nitrocellulose paper by capillary action. This type of blotting is time consuming and may take 1 to 2 days. For fast and more efficient transfer of desired protein from the gel to nitrocellulose paper, electroblotting can be used here. In electroblotting, nitrocellulose membrane is sandwiched between gel and cassette of filter paper, and then electric current is passed through the gel, causing the transfer of protein to the membrane. Here you can see the image of Western blotting method. The next step is blocking. Blocking is very important step in Western blotting. Antibodies are also proteins, so they are likely to bind the nitrocellulose paper. So before adding the primary antibody, the membrane is non-specifically saturated or masked by using casein or bovine serum albumin. The fifth step is treatment with primary antibody. The primary antibody is specific to desired protein, so it forms antigen-antibody complex. The next step is treatment with secondary antibody. The secondary antibody is enzyme labeled. So for example, alkaline phosphatase or horseradish peroxidase is labeled with secondary antibody. Secondary antibody is antibody against primary antibody, that is anti-antibody, so it can bind with antigen-antibody complex. The last step is treatment with suitable substrate. To visualize the enzyme action, the reaction mixture is incubated with specific substrate. The enzyme converts the substrate to give visible colored product, so band of color can be visualized in the membrane. Western blotting is also a quantitative test to determine the amount of protein in sample. Here in the image you can see the target protein, primary antibody, secondary antibody, enzyme conjugate, substrate, product and emitted light, which have all been represented in different colors. Application of Western blotting. To determine the size and amount of protein in given sample. Disease diagnosis detects antibody against virus or bacteria in serum. Western blotting technique is the confirmatory test for HIV. It detects anti-HIV antibody in patient serum. Useful to detect defective proteins, for example, Creutzfeldt disease. Definitive test for Creutzfeldt-Jakob disease, Lyme disease, hepatitis B and herpes. Thank you.